Today, the presentation will be focused on pharmacoepies. Mainly, we'll talk about the comparative safety and effectiveness research in the evaluation of, and its contribution to the evaluation of anticoagulant therapy. So these are my funding disclosures. So this is a brief presentation outline. So at the beginning of this presentation, we'll compare two types of the most commonly used evidence for the evaluation of treatment efficacy, effectiveness, and safety. So mainly, we will compare randomized clinical trials versus real-world evidence. And then after that brief introduction, we'll move to cover a few examples of studies evaluating anticoagulants using real-world data. So let's start with the first portion. So the goal of this presentation is not to convince you that the evidence we obtain from real-world data is better, worse, or equal to the evidence we obtain from randomized clinical trials. In fact, the evidence we obtain from real-world data in most cases can be complementary to the evidence we obtain from randomized clinical trials, especially in cases where data from RCT can be not possible or not even feasible. So I'm sure most of you are familiar with randomized clinical trials, but briefly, in a randomized clinical trial, patients are randomly assigned to the treatment or to the placebo group. Now, the random assignment itself ensures an equal distribution of baseline characteristics and disease risk factors between the treated and the untreated. So again, if randomization was successful, then one can compare the incidence of the outcome between the treated and the untreated without any further adjustment. Now, although randomized clinical trials are the gold standard for establishing treatment efficacy and safety, they have many drawbacks. Most patients who are recruited for inclusion in randomized clinical trials, they do not represent those we see in routine clinical practice. We have underrepresentation of patients with multiple chronic conditions. For example, patients with atrial fibrillation and valvular heart diseases tend to be underrepresented or excluded from drug approval trials. We have underrepresentation of the elderly or the frail population. And in many cases, it can be unethical or not possible to conduct randomized clinical trials in many populations, such as, for example, pregnant women and children. As you all know, randomized clinical trials are costly and can be hindered by slow recruitment. In fact, the cost of conducting one randomized clinical trial can be several $1 million. So given these limitations of randomized clinical trials, there is a wide interest in real-world evidence. And in this presentation, we're focusing on real-world evidence or observational research as it pertains to medications research. So here we have the definition of real-world evidence as it's defined by the Food and Drug Administration. So it's defined as data regarding the use, potential benefits or risk of a drug derived from sources other than traditional clinical trials. So within the pharmacoepidemiology world, we have two different types of data. We have governmental or commercial data. In terms of governmental, we have Medicaid and Medicare. And a few examples of commercial data, we have MarketScan and Optum. Now, regardless of which data framework you're using, within each data framework, we have different files. So for example, we have enrollment files, which tells you when did the patient enter the healthcare plan and when did they exit. We have outpatient claims. We have all of their diagnoses, procedures that were done in the outpatient setting. We have also inpatient data, meaning the date of hospitalization, date of discharge, diagnosis, procedures, and so on. We have outpatient prescription claims, including refill date, quantity, and strength, and so on. Now, the availability of laboratory data and death records tend to be limited. So for some of the data framework that I just mentioned, for example, for Optum data, we often have access on lab data for 20% of the sample. And death records, in most cases, cannot be obtained with commercial data, but we can obtain them from governmental data by linking the data to death records using Social Security numbers. So if we would look in any of the files that I just mentioned, we will see that we have multiple rows per patient. So here, this is a unique patient identifier within the data because the data has been de-identified. So we can see here, this is the same patient with 10 or 20 records within each of the files. We can see here date of birth, admission date, different diagnoses. And if we would stack all the patients, the million of patients within the data, you will get millions of lines. So at the end, claims data include thousands to millions of patient records, but we have to remember that those are primarily collected for billing purposes, which is unlike randomized clinical trial where the investigator have control over the variables collected in claims data. We have to use whatever is available there because the data were collected for other purposes other than research. So now what we just covered real world data briefly. So we'll move to the study that we will focus our presentation today on. So we'll talk about the risk of recurrent VTE and bleeding with apixaban versus rivaroxaban. All data, information, ICD-10 and codes are published in the appendix in our study in Annals of Internal Medicine if you need more data. So we'll start briefly by covering what was the motivation behind the study. So as most of you know, venous thromboembolism or VTE affect about 100% per 100,000 each year. This is a population at high burden, and one of the issues within the VTE population is that patients are at higher risk of developing recurrent VTE event within the first few months to a few years following their diagnosis. Now, in terms of available treatments, guidelines recommend the use of anticoagulants. So we have two different classes. We have warfarin, and we have direct oral anticoagulants, including apixaban, rivaroxaban, dabigatran, and edoxaban. What we know prior to publishing this study, so this was published, I think, in 2020, is that based on a clinical trial, Dawwas were found to be effective and safe when compared to warfarin. Now, the contribution of real-world evidence in this case is, as most of you know, when manufacturers are asked to prove that a drug is efficacious and safe, they are asked to compare a drug to a placebo. But when it comes to routine clinical practice, for example, in this case, under Dawwas, we have 4 different agents, and among the Dawwas, apixaban and rivaroxaban are the most commonly used agent. However, there is lack of head-to-head comparison between apixaban and rivaroxaban in clinical practice. So when clinicians have to select between the two, there is almost no data available to guide the treatment selection. So moving to the study design. So this was a retrospective cohort new user design, and we used data from Optum Clinoformatic, which is a commercial data within the US. I'll try to simplify this as I can. So the population included in the study, we included patients who had VTE diagnosis. So this was their incident VTE or first VTE diagnosis. And we used previously validated ICD-9 and ICD-10 codes to define VTE. We required patients to have initiated treatment with apixaban or rivaroxaban within 30 days of their VTE diagnosis. So they were required to be new users of either apixaban or rivaroxaban. And the day of initiation of either apixaban or rivaroxaban was assigned as the cohort entry date. Now, from that date, we required patients to have 12 months of continuous enrollment in medical and pharmacy benefit. And as you can see here, the 12 months was used to define baseline covariates, to look for prior drug use, and to apply exclusion criteria. So we restricted the cohort to patients who were 18 years or older, so anyone younger than 18 was excluded. Those without 12 months of continuous eligibility were excluded, and anyone who used anticoagulant for any other diagnosis during the baseline period were excluded because we wanted to focus on the new users of the study drugs. Now, from that point, we started the follow-up. So our survival analysis started from here. So patients would contribute person time as long as they continue to refill their prescriptions, allowing 7 days between refills. And I'll explain in the next slide exposure definition. And the follow-up would start at the occurrence of outcome of interest, which we will also explain in detail in the following slides. If the patient stopped or discontinued the medication, so for example, a new user of apixaban, stop apixaban, or same for rivaroxaban. If they started the study comparator, so for example, a new user of apixaban later in the study would start rivaroxaban, that patient would no longer contribute person time. At end of enrollment, meaning if they discontinue enrollment in their healthcare plan, and at the end of the study period, and in this case, we had data available until June 30th, 2020. And I'll stop before moving to the next slide and ask if anyone has any questions. Any questions about the design? No? Okay. So this is a summary of all ICD-9 and 9 codes and different study inclusion and exclusion criteria. You can find it online, so we'll move to exposure definition. So as I mentioned, patient would be considered exposed in the study if they started apixaban and rivaroxaban, allowing a 7 days or less between continuous refills. So just to show you how does the different scenario play within the data, because you will see things sometimes that are unexpected. So let's start with the first patient here. So this patient initiated treatment with apixaban. So the patient was included in the study. And we can see that the gap between the different refills was 7 days or less. So this patient was continuously exposed until the end of the study. For patient number 2, this patient initiated treatment with rivaroxaban and continued exposure without any gap. So this patient was considered continuously exposed until the end of the study. Now, if we look at patient number 3 and patient number 4, both of those patients were not actually included in the study because they used anticoagulants during the baseline period for other diagnoses. So we didn't include them because we wanted to focus on the new users of the study drugs. Patient number 5, 5 days included and also continuously exposed. Now for patient number 6, this patient was included in the study. However, if we look here, we can see that the gap between refills was 60 days, which is more than 7 days. So this patient was followed from this point, and then we no longer contributed person time prior to the 60 days because the gap was more than 7 days. Now, patient number 7 is a rare case. So this patient initiated treatment with apixaban and rivaroxaban on the same date. So this patient was excluded. And lastly, we have this patient number 8 initiated treatment with the rivaroxaban. However, during the study period, they also added apixaban. So at this point, this patient no longer contributed person time in the study. So for outcome ascertainment, the primary effectiveness outcome was recurrent VTE event. And this was defined as a composite of deep vein thrombosis and pulmonary embolism. In most claims data, we used validated ICD-9 and ICD-10 codes, and we restricted the definition to those listed in the primary or first listed position. A common side effect of anticoagulant is bleeding. So our safety outcome was bleeding event, and this was defined as a composite of intracranial and gastrointestinal bleeding, similarly defined based on ICD-9 and ICD-10 codes. Listed in inpatient claims in the first or principal position. And a question we had from reviewers is why we didn't include, I'm sorry, we didn't include bleeding event documented in outpatient setting. And the reason was that most bleeding event in claims data that are recorded in outpatient setting has low positive predictive value. So we try to examine them in a sensitivity analysis, but not in the primary analysis. So for confounder adjustment, we included a very large number of confounders. So included demographics, risk factors for VTE, risk factors for bleeding. We also examined baseline medications used. And lastly, we included measures of intensity of healthcare utilization. For example, total number of hospitalization within the prior year, total number of outpatient visits, or total number of prescriptions within the previous year. And we also use previously validated definitions for confounders. Now for adjustment, we used a propensity score, which is a commonly used method for confounder adjustment in pharmacoepi studies. And we used one-to-one matching. And we matched patients based on a caliber width of 0.1 of standard deviation, a flowjet of propensity score. And I'll explain briefly propensity score in the next 2 slides. We use standardized differences to compare the balance before and after matching. And we calculated the incidence rate of the effectiveness and safety outcomes and reported them in our analysis per 100% years. We estimated the marginal hazard ratio and corresponding 95% confidence interval using Cox proportional hazard regression model, and we used robust variance estimator to account for the matching nature of the sample while adjusting for calendar year. And we use also a Schoenfeld residual in order to check our survival assumption that we met them. Can I ask how many people in the room are familiar with propensity score? Anyone? 1, 2, 3. Okay. Okay, I'll try to simplify it. So how do we build a propensity score model in any data? So using propensity score does not restrict itself to pharmacoepidemiology study. It can be used in any research question if you have a very large number of variables and a very small number of outcomes. Because if you would adjust for all of those variables in your regression model, your model become unstable. So one way to overcome this issue is to use propensity score, which is a summary score for, I mean, depending, I think in this study we had 41 or 42 variables. So the way to build a propensity score in claims data or in any other data, if you recall when I showed how does claims data look like, we had multiple rows per each patient. So the end table to, do you want to guys get a pizza? Everyone's looking at the pizza. I'll continue talking. You can go. You can feel free to go grab a pizza. So the end component or your end table prior to running a propensity score model, you need 1 row per each patient. So the first step is to identify all the confounders that you want to include in your model. So let's assume we have 9 different variables. So for patient ID number 1, this patient initiated treatment with apixaban. So this patient get 1 here. And then for each of the different comorbidity, I want to code 1 and 0. And then for patient number 2, this is a rivaroxaban user. I coded for all the different variables in 1 row using all of the different data domains that I have, again, as a 1 or 0. So after I created this table, I want to use logistic regression model to estimate my propensity score. And why we're doing this? Because propensity score is the probability of initiating treatment with one exposure versus the other as a function of all of the different covariates that I have in my study. So propensity score by using logistic regression model is in fact a probability. So it's a number between 0 and 1. So after you plug in all of your variables, similar to what we're doing here, regardless of how many variables you have, you will end up with only 1 number per each patient. So if we look at the table here, using the logistic regression model that I have showed, the model outputted a propensity score for each patient. So this patient has a propensity score of 0.01, 0.20, and so on. So after I obtain my propensity score, I have to decide how I'm using propensity score. And a commonly used method is matching. So I would tell the software to match patient based on a caliber width of, let's say, 0.2. So if this patient is 0.20, let's say match this patient from the rivaroxaban group to a patient from the apixaban group who have a propensity score of 0.22 and so on. So the software would keep matching the patient until there is no longer any more matches, which brings a disadvantage of propensity score and the question that we would always get. So what happens to the unmatched population? And we will address this later. And I mean, the benefit of propensity score, if you would compare an observational study that used propensity score matching to a randomized clinical trial, you would achieve what we call in the observational data world a hypothetical randomization. So all of your baseline characteristic would be similar if matching was successful between the treated and the untreated or between the 2 groups that you are comparing. So moving back to our study, so using Optum data, we identified an incident VTE cohort of 260,622 patients. And among them, we ended up with 89,292 patients. So we applied all of the exclusion criteria that we listed earlier. So our cohort included 49,900 patients, and this is prior to propensity score matching. We can see the numbers. We have 28,287 apixaban users versus 21,613 rivaroxaban users. So in this study, we used 1-to-1 matching, and this is why our numbers are exactly the same between the apixaban and rivaroxaban users. I mean, it's possible to, if you have, let's say, the proportion of apixaban users is much larger than rivaroxaban users. If you have 10 times in one arm versus one time in one arm, you could do 10-to-1 matching or 1-to-10 matching and so on. So here we did in the primary analysis only 1-to-1 matching. So this figure, we created this figure based on the data we have, and we have here the calendar year. So we looked at data from 2015 to 2019 And we can see here on the y-axis, we have patient initiating anticoagulation. We have apixaban, the blue line here. The gray line is rivaroxaban, and we have warfarin here. So our conclusion from this figure is actually consistent with other studies in other patient population. We can see that apixaban and rivaroxaban are still the most commonly used anticoagulant within the VTE population. Warfarin use continued to decline across the study period, and this is related to many disadvantages of warfarin, including the need for frequent laboratory monitoring and many drug and drug-food interactions. I mean, dabigatran and edoxaban are of limited use, and this is consistent also with other patient population. So moving to table 1, so I'm going to only highlight a few characteristics just for the sake of time. So here on the left-hand side, this is our pre-matching cohort. So we can see here we have our apixaban users, 28,000 versus 21,000 rivaroxaban users. So patients within the apixaban arm were slightly older. We can see the standardized difference is more than 10%, suggesting that there is some imbalance here. However, within the post-matching cohort, the standardized difference was no longer significant, suggesting that with the use of propensity score matching, we achieved adequate balance. Similarly, patients within the apixaban group had higher prevalence of comorbidities, including chronic kidney disease, chronic lung disease. We can see 41% versus 28%. And for chronic lung disease, we have 36% versus 30%. And again, those baseline characteristics were no longer significant after matching. I mean, without going further in all other comorbidities, we can see the balance here in the post-matching cohort. And similarly, patients who were in the apixaban arm tend to have higher use of angiotensin receptor blockers, 22% versus 17%, beta blocker, 34% versus 25%. And again, we can see the balance in all baseline medications and measures of healthcare use in the post-matching cohort. I know that this figure is very small, so if you want to simplify, if you ever did a propensity score matching, and if you want to simplify table 1, one way to do so is to draw something that we called a love plot. So here on the y-axis, we have absolute standardized difference for my study. And we can see the line here where 10% suggests the line where more than this, there is imbalance, and less than this, we have an adequate balance. We have listed here all the comorbidities and medications that we included in our propensity score model. And you can see that the black dots here are the pre-matching cohort, whereas the yellow dots are our post-matching cohort. And what you want to see in this figure is that everything from the right side, when you do the matching, you need to move to the left side. So we can see all of the orange dots here are below 10%. Suggesting that by using propensity score matching, we achieved adequate balance in all variables. So we'll move to the study results. So after we did our propensity score model and we ran our Cox proportional hazard model, we found that use of apixaban versus rivaroxaban was associated with a lower risk of recurrent VTE. And we can see the hazard ratio of 0.77 and the confidence interval of 0.69 to 0.87. Similarly, use of apixaban versus rivaroxaban was associated with a lower risk of the composite of bleeding event. And we can see the hazard ratio of 0.60 and the confidence interval of 0.53 to 0.69. So we stratified the primary analysis to the individual component. So recurrent VTE, we stratified that to deep vein thrombosis and pulmonary embolism. And we can see we have consistent findings within the secondary analysis. And for bleeding, we also stratified the analysis to GI and intracranial bleeding. We can see the estimates are consistent. However, the confidence interval here for intracranial bleeding crossed the null just because of the small number of events for that outcome. So in any pharmacoepidemiology study, you have to make many assumptions. And the idea is that when you go to your sensitivity analysis, you have to challenge all the assumptions that you made. So one assumption, if you recall, in our exposure definition, we considered patient exposed as long as they continue to refill their medications, allowing a 7 days gap or less between continuous refills. So we challenged this assumption by changing the gap to allowing 15 days and 30 days. Now, an issue that sometimes comes when you run any propensity score model, you can plot a histogram. So an issue that you sometimes might see that there is extreme observations at the tail. And one way to address this issue is to trim propensity score tails. So we did several methods to trim the tails of propensity score that we will show in the next table. I mentioned that propensity score matching is often criticized because of the exclusion of the unmatched population, but there are other methods where you can include the whole population. For example, you can adjust for propensity score instead of matching. Instead of using one-to-one matching, we did a sensitivity analysis where we did one-to-one matching, where we tried to retain a larger number of users of apixaban and rivaroxaban. In our primary analysis, we included only recurrent VT event occurring in the inpatient setting. However, in a sensitivity analysis, we also accounted for those happening in the outpatient setting. We conducted a negative control outcome analysis in order to examine or to test for the presence of residual confounding. So for those of you who are not familiar with a negative control outcome, this is an outcome that doesn't have an established association with the exposure. And the idea, if I would conduct the analysis, that we would get a null finding. And lastly here, as I mentioned, in most claim-based analysis, we don't have access to many of socio-demographic factors and lab values. However, in Optum data, we had access for those variables only in a subset of the cohort. So what we did, we conducted the primary analysis without those variables, and we conducted a secondary analysis where we examined whether those variables were well balanced in the post-matching cohort, despite not including them in the primary analysis. And the assumption was that by using a new user active comparator design, we were hoping that we would obtain a balance despite not including those variables in the primary model. So we can see the results of the different sensitivity analysis. So the first few, we increased the permissible gap between refills from 7 days to 15 days, and from 7 days to 30 days. And we can see here the results for both the effectiveness and the safety outcome. So we have consistent results despite changing our study assumption. We trimmed the propensity score distribution tail to 95th and 5th percentile, and to 85th and 15th percentile, and lastly, to 75th and 25th percentile. And we can see the study estimate remain consistent for both effectiveness and safety. We adjusted for propensity score in the outcome model instead of matching, and we used one-to-one matching with replacement. We changed the caliber width. If you recall, we used the caliber width of 0.1 in the primary analysis. So we also challenged this assumption. So we used the caliber width of 0.2, 0.01, and 0.02. And we can see there was a slight changes in the number of apixaban and rivaroxaban users that were matched, but the study results also remain consistent for both effectiveness and safety. We included recurrent VT event occurring in the outpatient setting, and we can see also apixaban associated with lower risk of recurrent VTE. And lastly, our prostate and breast cancer negative control outcome resulted in a negative null finding as we were hoping. We conducted also several subgroup analysis to examine for the presence of effect modification within selected subgroups. And we can see all of the different groups here, for example, presence of cancer at the baseline period, use of non-steroidal anti-inflammatory drugs, use of antiplatelet, type of VTE, presence of mechanical heart valve, hip or knee replacement in the stage renal disease. And we reported the p-value for interaction for both recurrent VT and bleeding. Our interpretation of the result was done after we corrected for multiple testing using Banforoni correction. So the study results remain consistent within each of these subgroup analyses. So at the time we published this study, which was almost 2 years ago, the only study that examined apixaban and rivaroxaban was actually a meta-analysis of randomized clinical trials. And that analysis reported no difference in recurrent VTE with apixaban versus rivaroxaban. But we can see that the confidence interval for that meta-analysis was wide, and that our result almost and confidence interval falls within their confidence interval. Now, the same analysis produced similar findings for bleeding outcome. We can see from our study, our hazard ratio was 0.63 compared to a relative risk from that analysis of 0.69. And the only other study beside that meta-analysis was actually an earlier analysis that we did using market scan commercial data. However, we use an earlier years of the data. So we use data between 2013 to 15, and we reported similar findings. The only difference was that patients who are enrolled in the current study that we identified using Optum data, using later years, were slightly older and had higher prevalence of comorbidities, including some that I'm listing here, such as hyperlipidemia, chronic kidney disease, and liver disease, which suggests that clinicians are feeling more comfortable with apixaban, that they're prescribing apixaban to a sicker population. Now, in terms of limitation, most claims data do not provide information on lifestyle variables such as body mass index and smoking. We also have limited information on over-the-counter medications, which suggests that it's possibly that there is a slight impact of unmeasured confounders on this study. However, to address this concern, as I mentioned, we took a proportion of patients where we had access to their lab values, and we did not include those lab values in the primary analysis. And what we found that by using a new user active comparator design, in fact, created an adequate balance in the post-matching cohort in several laboratory measures, including creatinine, cholesterol, triglyceride, and hemoglobin A1C. And the results that we showed in this secondary analysis was actually confirmed by another analysis, which reported that linked claims database to electronic health record found that new user active comparator design led to balance on several variables not available from claims data. That analysis looked at prescriber specialty, aspirin use, which is often used over the counter, body mass index, and alcohol use. There is the potential for outcome and exposure misclassification, and this is why we challenged the definitions in our sensitivity analysis. In terms of generalizability, since the data was restricted to patients with commercial healthcare coverage, the result would be generalizable to that population, and that would limit the generalizability to patients with governmental health insurance or the uninsured population. And lastly, we mentioned that propensity score matching lead to loss of participant. And one way to address this is to conduct or use different adjustment method in order to look at the original sample that didn't exclude the unmatched population. So in conclusion, in this comparative effectiveness and safety study using real-world data, adults with VTE who initiated apixaban had a lower rate of recurrent VTE, intracranial and gastrointestinal bleeding compared with rivaroxaban. And we were hoping that the findings from this study would guide clinicians and patients regarding the selection of anticoagulant for treatment with VTE. So the story of the study didn't end when we published the study. In fact, this was published, I think, in October of 2020. And 1 month after we published the study in Annals of Internal Medicine, CVS, Caremark, which is part of CVS, decided to exclude apixaban from their formulary list, citing the absence of evidence. So representatives from the American College of Cardiology and American Society of Hematology took this study, which was published in Annals, and said, here is the evidence that apixaban is more effective than rivaroxaban. However, CVS argued that our study included patients who have dual enrollment in Medicare, making that population not applicable to patients who receive coverage from CVS. They also said that the study did not look at effect modification, meaning that whether the study would apply to the older and the younger population. So it was Christmas time, and representative from patient advocacy group. I mean, patients were reporting this all over the US, being denied apixaban prescription, which is very expensive. So it was Christmas 2020. So they reached out and asked whether would be willing to replicate the study. So what we did, we said, okay, we'll answer CVS questions. So we replicated the same analysis, excluding patients who have dual enrollment in Medicare. And we looked, we looked at effect modification by age. And what we did, we published the study results within the American Journal of Hematology. And this was a response to CVS formulary changes. And so, as you can see, when we excluded patients who have dual enrollment in Medicare, our results remain consistent for both recurrent VTE and bleeding. We also, to answer CVS question, we looked at effect modification by age. So we wanted to prove, okay, the study results apply to the older and to the younger age group. And as you can see here, p-value for interaction were not significant, suggesting, in fact, that there is no difference between the elderly and the younger population. We published a study, CVS reverses decision, and apixaban was back to the formulary list. So this study was actually, in fact, the first time that we have seen in our group a pharmacoepi study make a health policy decision. So currently, in terms of anticoagulant work, we're looking at anticoagulant within stroke and inflammatory bowel disease patients, and those funded by NHLBI and the American Society of Hematology. We also expanded the work to patients with atrial fibrillation and acute coronary syndrome, patients with atrial fibrillation and valvular heart diseases. And we also have another line of research looking at diabetes drug and anti-obesity medications that is parallel to this work. I mean, we have pending funding for the other line, but this is the currently funded work. I've been grateful to work with a very large number of mentors who shaped my career. I'm listing a few here. There are many more. I want to also acknowledge my postdoc who joined us from the University of Colorado.